Greetings, ladies and gentlemen. This is Shofiul from the Department of Zoology, University of Dhaka, Bangladesh. I'd like to welcome you all to hear about our work, ATP-binding cassette transporters in Tephritid fruit flies. In 2022, the global industry of agriculture owed more than 12 trillion US dollars, and while the fruit and vegetable industry owed more than 2 trillion US dollars. Millions of people depend on these two industries, and they are highly significant for the global food security. But there are several major challenges are present, and agriculture pests are the biggest ones. So when we are talking about agriculture space, we must address the biggest threat, Tephritid fruit flies. So what are they? Tephritid fruit flies are one of the most common, most destructive, and highly invasive species throughout the world. Till 2018, more than 4,000 species of Tephritid fruit flies has been identified by the scientists. Alone, the Queensland fruit fly, Bactroceras trioni, causes the economic losses of 300 million US dollar losses in the Australian agriculture industry. Also, the Mediterranean fruit fly, Tephritid capitata, causes about 12 billion euros of losses both in Europe and Asia. But numerous biological control measures are undergoing right now, and a lot of them are highly focused in ABC transporters of Tephritid fruit flies. So, what are ABC transporters? ABC transporters or ATP-binding cassette transporters are one of the largest superfamily and one of the oldest superfamily of protein, which are found in all the living organisms. They are also the large superfamily of diverse membrane protein, and they utilize the energy of ATP hydrolysis to transport the solutes across the membranes. Also, they are involved in various biological processes which are important for any species, such as nutrient uptake, multi-drug resistance, heavy metal tolerance, detoxification, insecticide tolerance. And as a result, these proteins show very important aspect, and they can be targeted as a pest control and resistance management process. So what we are trying to do in our study, we try to do some existing literature review on ABC transporters, giving a special emphasis on Drosophila melanogaster, as it is one of the highly studied organisms throughout the world. Then we evaluated the available NCBI resources for Tephritid fruit fly proteomes. We have done ortholog-based search for Drosophila melanogaster ABC transporters proteins, which are available in the available Tephritid fruit fly proteomes to identify the proteins and to comparative and phylogenetic analysis. And also we have done ortholog-based characterization to identify important proteins for future biological control application. So for our study, at the very first part, we went to the Flybase website and we have downloaded the ABC transport protein sequences. We have found 145 ABC transporters from 56 genes, which are divided in 8 different subfamilies. Then we went to the NCBI website to download the available Tephritid fruit fly proteomes. We have found 8 species for the proteomes. We have found Bactroceras dorsalis, Bactroceras latifrons, Bactroceras oli, Bactroceras trioni, Zeguracus cucurbiti, Rhagoletis promonella, Rhagoletis zephyria, and Tephritid capitata. After that, we use BLAST-P mechanism to identify and do our ortholog-based search, and we analyze the data that we found from the BLAST, and then we use reciprocal BLAST to confirm our hit that we have found. After that, the proteins and the genes that we have found, we have divided them in their subfamilies, and after that we compare our findings of proteins with species to species one another. So what we have found in our study. So our orthology-based search have found 67 Drosophila melanogaster ABC proteins across these Tephritid fruit fly proteomes. Unfortunately, we couldn't find 78 proteins, and there was no hit for them. We have found 43 ABC transporters for Bactroceras dorsalis, also 50 transporters for Bactroceras trioni. 49 transporters for Bacteria latifrons. Both Regulatis pomolella and Regulatis zephyrida have 48 ABC transporters. Ceratidis capitata have 56 transporters. Interesting fact is that we have found 48 ABC transporters in Bacteria oli, and we have found 30 novel ABC transporters among these 48. We have also found 56 ABC transporters in Zygotococcus cucurbiti, and we can say that we have found 8 novel ABC transporters in them too. So after that, we categorize our protein findings in 8 different subfamilies of ABC transporters. And we have found that most of the proteins of ABC-B subfamily and few proteins from ABC-E subfamily and ABC-F subfamily are conserved. But we have found a wide diversity in subfamily ABC-C proteins. And our phylogenetic analysis showed that within Bacteroceras genus, the hits that we have get from the blast and reciprocal blast remain more or less constant. But significant fact is that we have found a remarkable amount of similarities in both Zygotococcus cucurbiti and Ceratidis capitata ATP-binding cassette transporters, which are more or less similar in number, and they are also similar in when we are divided them in subfamilies. So another fact is that we have found some highly important proteins across all the Tephritid fruit fly proteomes. We have found multi-drug resistant 49, multi-drug resistant for 50, and multi-drug resistant like protein 1, which is highly important for multi-drug resistance mechanism. We have found heavy metal tolerance factor 1, which is important for heavy metal tolerance. We have found red dog mine protein, which is important for zinc detoxification. And we can say that these proteins are highly important for the mechanism and for the survival of this species. And these proteins can be utilized in future for the future pest control management. So if I want to summarize what we have done in our study, we can say that this is the first comprehensive study of ABC transporters in 8 different Tephritid fruit fly proteomes. We have found 67 different Tephritid fruit fly proteomes, which are ortholog to Drosophila melanogaster, and few ABC transporters in Zygotococcus cucurbiti and Bacteria oli. And as I said earlier, we have identified some important proteins which are important for multi-drug resistance, heavy metal tolerance, detoxification, and also insecticide resistance. So we can utilize these proteins for our future investigation to find a better biological control solution. And our current study is an important starting point for novel and unannotated ABC transporter in Tephritid fruit flies. So we can use them for multi-omics data set and explore the enormous possibility that we have to the pest control management. So in the end, I like to thank you all who have joined here us today. I like to thank my fellow co-author Khandakar Asif Ahmed, who helped me throughout the journey in every best possible way. And I also like to thank the organizers for organizing this beautiful program. The Tephritid fruit flies are highly invasive, and this species must be controlled for our global food security. And again, we can create a better world together if we work together. Thanks so much.
Thank you very much, Shofiul. That was a really interesting talk. To everybody in the audience, if you have any questions, you can put them in the Q&A. In the meantime, Shofiul, if you don't mind, I had a question.
Yeah,
so
sure.
You mentioned that you identified some important transporters, but how do you define the important ones? Did you verify them through literature study, or how did you define the important ones?
For this part, as I said earlier, we have did some literature review on for the for Drosophila melanogaster ABC transporters, and we have studied them thoroughly so that we can identify those proteins which are important for the pest control management. And we also have some special papers we saw them, and we have find some remarkable how can I say, remarkable results, which are currently worked by the scientists all over the world, which are doing some expert works on these proteins. And these proteins show high amount of potentiality to do some very important pest management control. So, and also these parts, their significance are also annotated. So I think with those previous literatures and with those previous parts, we identified those important proteins, and we tried to characterize these proteins that we have found in our study and tried to compare them that yes, we have found them.
Okay, thank you. Thank you very much.
Thank you.
So we still don't have any more questions from the audience. So I'll just have another one, another
Sure,
small
sure.
question. So
Sure.
towards the end, you mentioned that we could use multi-omics data sets to look at more transporters.
Yes.
So what kind of multi-omics data sets do you have in mind, and what omic modalities are you planning to look at?
How can I say? Right now we have only 8 proteomes which are available in the NCBI dataset right now. Even though there are more than 13 species has been done with whole genome sequencing, but we have only 8. We currently use the Drosophila melanogaster ABC transporter orthologs to find out our data, but I think in future we can use publicly available RNA-seq data, and also we can utilize the several aspects and several development stages of this species to identify the proteins which are active in their several life stages. And I think by this process, I think using RNA-seq, then chromosome mapping, and complete annotation of their chromosomes can be highly helpful for us to control these things. And I think in my mind, I think I'm going to use RNA-seq data and several other methods that we can utilize in future, I think.